Thank you very much. I realize that probably my only credential to be invited is my common nationality with Leonardo. And now I realize maybe Uli had some other thinking as well in doing this crazy idea to get me to cover for this talk. I realize that a lot of what we've been discussing today with all our speakers actually has a very profound relevance to the fundamental question that we have in research in biopharma, that is how we sustain innovation in a world that is clearly the expectations are much higher. I'm not going to say on the 500th anniversary to Leonardo, you had it easy, that the expectations of your time were not as high as they are today. But in some way, the common theme of Leonardo is that in a single person, you have this multidisciplinary invention and creativity that is probably very much applying to what we have to do today to remain relevant in the innovation in biopharma. In a single person, you had physicists, humanists, artists, anatomists, and many, many other things. And he could do that in isolation, as very nicely described by our speaker. But I think for today, for the biopharma innovation to still remain vibrant, we have to do a lot more than what we have done in the past. And you will see some common themes. I always like to start with the fundamental. I'm a physician, and the fundamental reason we're all here in this room is that at the end of the day, our vision is that we are a science-driven organization delivering transformative medicine that make a real difference in patients' life. And this seems a very simple statement, but it's becoming very, very difficult. If you think about the various drugs that are approved in different countries today, how many of those drugs are truly transformative and truly make a real difference in patients' life? And a lot of this has to do with what we discussed throughout the day in some of the other talk. We have to start from the patient, really be at the center of what we do. We need to leave not just a bench to bedside, but also a bedside to bench. We need to learn from human biology, from the gap in current therapy, from all the technology that allow us to understand the human body and how the disease develop in order to inform our powerful discovery engine. And we need to focus on mastering the depth of human biology. I will go into some of these topics a little bit more. There are some other very common themes. So even starting from our ambition and vision, I think you can see that the bar is very, very high. Just a molecule, a good target, and a good development plan is not enough today. We have to do a lot more. We have to go around all the different tools to make understanding what is the best way to address the particular patients. And I very much agree with the team that the disease, as we described today, is probably very heterogeneous. It's a conglomerate of multiple different genetic predisposition, epigenetic phenomenon that in each individual is probably somewhat different. And we have to start learning that complexity. I want to go back to basic. I think this was already expressed in a different way. But why is it not sustainable to continue doing the brutal force that has been sustaining pharmaceutical industry for many, many years? It's on the left. The cost of developing new drugs, including the cost of failure, it's massively increasing. And probably the $2.6 billion, it's from 2014. It's getting even higher today. And you can see the escalation on the left. In the same time, the number of approved drugs in different countries is actually pretty much flat. But I would add also that of the drug approved, the ones that are a market success because they're making a real difference in patients' life are more and more a minority. So the approval is not even the real parameter to look at. It's the one that really make a difference. Now, we heard early on from Uli the miracle of immuno-oncology, but these kind of things probably happen only once every decade, and I think they are so few, and it's just the beginning of our learning curve, even in fields like immuno-oncology. So this is not sustainable, even from a financial point of view. When I say brutal force, it's to taking a 95% basically failure rate from first in human all the way to registration of a new drug, that whether or not it's making a real difference is also a question. So this is a very simple consideration, but the traditional pharma model was enough, frankly was enough even having some empirical observation in a plant extract or something like that. But in general, in recent years, was enough to identifying a target. With our chemistry, we were able to develop a very, very good molecule that will modulate that target, then have a clinical development program, and in a very linear fashion, then put it on the market and be able to sell, and making difference in that way. Frankly, this is not sufficient anymore. If you don't really understand in depth all the disease biology around that target, develop a biomarker, understanding the special segment of the population affected by that pathology, and particularly the particular pathways that you're looking at. If you don't understand how the disease is providing specific gap in the therapy that you're trying to address, and you go even in some value-based proposal for the patient benefit, I think if you don't increase your reach into all this environment, I think you have very little hope to develop really innovative therapy. Some examples are going to be shown in the next few slides. I want to make another point at the macro level for the industry that many of you are aware of, but it's very important to make this. If we look at innovation as the origination of the patent, so where the real innovation is coming from, you see in yellow the very large pharma that we identify with. And actually, the percent of patent originated directly from large pharma is actually going down. Mid-size perhaps going up, and small is definitely growing. But most important, if we look at the evolution from where you start, that is the innovation, the patent origination, where is the drug registered, the middle bar, and where is marketed, that is the last bar, you can see that the large pharma is more and more growing from when you go from origination of the ideas and the patent into the market. In other words, it's really the small organizations that are taking risk and they're really trying to innovate in more profound fashion that in blue are generating the origination idea, and then they are bought over time by big pharma. Mid-sized pharma looks a little bit in the middle because they keep their innovation and they contribute in a similar fashion to the market. It's an interesting point of view that is not just the general view how much more innovation is required, but the source of innovation is quite different even compared to a few years ago. This is just to reiterate the fundamental issue that just one molecule, one target, and a good development program is not going to make it anymore. You really have to have a full understanding of the biology around that, from a predictive value, the diagnostic, understanding the segment, being able to monitor this over time, having pharmacodynamic markers, to understand the specific targeting for the patient, the subgroup within the disease that you want to go after. And that's clearly also illustrated by the power that we have with more modern technology to really gather data from population-based information or even from biology, like we do in screening of genomic data, and really reconstruct that into the strategy for discovery. This is just a quick example that is applying also to the preventive nature of the disease, is liquid biopsy. You can take advantage of the fact that, for instance, tumor cell shed DNA, but also cells or cell-free RNA that can be sampled from the bloodstream. And through this, you can have a signature genomic picture of the tumor in the lung or any other site where this DNA is coming from. So non-invasively, you can phenotype the tumor at the genomic level and try to find the right therapy for the right patient. This is technology we're using right now for our tepotinib filing, a c-Met inhibitor. The complexity of the molecule, you remember, it mostly was medicinal chemistry, and that's still great. When you couple medicinal chemistry with fantastic biomarker data, genomic data, you can have precision medicine at its best. But we can also target specific targets within the tumor or within a disease by coupling a small molecule or other antibody that have multiple functions to targeting different antigens. So you can have the example monoclonal antibody, but also therapeutic protein, fusion protein, antibody-drug conjugates, in which you're really coupling the antibody in a way that is driving wherever molecule to very specific subgroup of patients that express a certain antigen. So this concept of complexity of the molecule is also linked to the idea that we want to have the right therapy for the right patients. These are just examples how the drug industry has to continue progress and modify. If you stay still and continue the linear thinking of the past, it's almost impossible to continue dealing with that 2.7 billion and plus of cost of failure that we're talking about and making a real difference in patients' life. This is my last slide, because I see that the clock coming down, and it's very similar to the concept expressed by many of you. We have to start from the patient. The patient is a role not just in understanding the human biology in depth, so study each patient with all the different technology available to us, from new generation genome screening to artificial intelligence and other approaches, fully understand the patient in that way, but also fully understand the need of the patients. What are the stakeholders that are involved in determining how you're going to develop the drug for that specific patient? And having a constant feeling that a deep understanding of disease biology from human experience, understanding the diversity of each patient and how you can best serve that patient, it's going to generate the right patient, the right target, the right compound for the right treatment combination, and truly transform the life of the patients. So we in our company and others are really start to change completely the way we think about drug discovery and development, and put the patient at the beginning and the end of our journey to serve the patients in the future. Thank you.